The next topic is how do we produce this transgenic cattle. Now it is much similar. जैसे we have done in so many other cases, similar method is being followed. Just that in here, what we are going to do is instead of your mice, instead of mice model, we are going to take cattle over here. Okay, and the way of collecting eggs is different. How do we collect eggs is just by super ovulation. Okay, this is how we did in mice. We super ovulated the mice and we took out the eggs. But in here, what we will do is we will collect the oocytes, that is eggs, from the slaughterhouse. Why? Because after killing the animal, the egg is just going to go waste from its body. So we just collect those eggs. Okay. Now what we do, since the eggs have been collected, they have to be nurtured now. Okay. So we will nurture them using some media in the laboratory. So that is called in vitro maturation of oocytes. Oocyte as an egg. Then the next step is we will fertilize the egg. That is also in the laboratory in the test tube. So in vitro fertilization using a bull semen, because it's a cow's egg. So we will use a semen from bull. Okay, and then just to select the egg which has a proper pronuclei. If you recollect my the first lecture, that is DNA microinjection, you would remember the pronuclei was important. Okay, so what we will do here is we will centrifuge all the eggs that we have collected and we have fertilized. Why? Because when you centrifuge, what happens is to egg yolk hai, okay, the yolk will get concentrated. Why do we have to concentrate is that normal eggs mein the male pronuclei is not visible properly. So if you want to visualize the pronuclei properly under the microscope, then you have to centrifuge it so that the yolk gets concentrated at one place. So the yolk, because the yolk is kind of spread out in the egg, so we want to concentrate it. Okay. So that is the reason why we are going to centrifuge it. So as we see, the steps are, first is collection of oocytes, that is we will collect the eggs from the dead animal. Then it could be collected from the live animal also, just that it is one more way of collecting, that they can also be collected from a dead animal. Then if they are collecting from slaughterers, it's very easy because people do eat cow, we don't, but they do eat cow. And then we can also have eggs from the slaughterhouse. Okay. Then the next is we will mature it in the lab and fertilize it in the lab. Okay, then artificially we'll fertilize it, and then the next step is we will centrifuge the egg to concentrate the yolk. You should always remember why we are centrifuging, because so far we have seen so many methods, we did not centrifuge any egg. Okay, and here we are doing, because this is not a normal egg, this is the egg of a cow. Okay, the next is the next is producing microinjecting. First you will take the DNA. Okay, not we can also produce it recombinant DNA. Okay, and transfer that. But in case you don't have anything such that in mind, you can just take a normal DNA with a good feature, or you can also take a recombinant DNA, okay, and then microinject it into the male pronuclei. Okay, this is what we did in the DNA microinjection. We are doing that only here. Okay, but this one, since it is modified a bit, okay, like we have added centrifugation and all. So this method is called modified mouse transgenesis DNA microinjection, because initially we did just DNA microinjection. In here, what we have done is we have centrifuged. That's why this method is called modified mouse transgenesis DNA microinjection. This is the name of the method. Okay. So after we have injected the DNA into the male pronuclei, what we do is we again develop the embryo. We will let the embryo develop in the laboratory condition. Okay, because we want to know whether the cell is going to divide or not. Okay, until the cell has been reached to embryo stage, we are going to nurture it. And then what we will do is, like again, remember yesterday I told you like until blastocyst stage. So until that stage, I am going to develop it, and then I will implant this embryo into a foster mother, but without any surgical process. Okay, it is done with non-surgical. This is called non-surgical implantation. Now, how do we do this is in natural estrus. What is estrus is, please write down the meaning. Okay, I will also type it in the chat box. Estrus is a stage of menstrual cycle where you can implant the egg because the body is prepared for the, like it has developed the endometrium and it is prepared to receive the egg for development. Okay, estrus is a stage in the menstrual cycle. Okay, I'm just putting so you all can know. For development of, okay, so this is a stage, and then what we do is after its birth we will screen it using we can do PCR for the DNA analysis, anything to assay the DNA. So what we will do is we will screen the DNA and check if the transgene is there in the offspring or not. Okay, so basically this is a similar process, just that there are few changes here and there. So first is concentrating the yolk, and then we go for in vitro development of embryo, which is already seen, and then we non-surgically implant the baby. Okay, that is implant the embryo. Is this clear about how we produce transgenic cattle? Okay, so just